Yes. Um, so maybe I'll just share my screen and with my details, I'll also introduce myself then and there. Okay. Maybe I'll share my screen so that it is easier to switch between presentations, right? Okay, so some of your peers are actually still joining, which is why I wanted to wait in the beginning. Um, okay, I'll share again, okay? Yeah. So in case you have a doubt between the session, you want to ask something, you want a little more emphasis to be laid on some specific point, or any other interruption with the meeting that is going on, you are free to turn on your mics and interrupt me. That is all right by me. Um, as for these meetings, so for people who have attended other NPTEL courses, this may be a regular thing. For people who are new, there are these NPTEL courses that are there on the website, and you are attending them. A lot of these courses are actually reruns of the pre-recorded courses, right? So, although there are professors who do take live sessions, even if it is a pre-recorded course, but most of the pre-recorded courses runs by itself, and then there are teaching assistants that are there to help you. Right, so there are your teaching assistants, Smith and Webby, I believe, who have introduced themselves to you also in the discussion link. And then there is me, who will be conducting these live sessions, right? So that we have a platform wherein, there is live interaction between us, so that if there is any point that you are not understanding in the presentations, you are free to discuss it over here. Also, a part of these live sessions will be to discuss the assignments of that week also, right? So when the assignment is actually ongoing, we'll not discuss it because we do not want to disclose answers of ongoing assignments. But I believe your week one assignment is already over, so we are free to discuss that now, right? So what we'll do is we'll split these meetings into two parts. One of them will be this discussion of assignments, the previous year assignments, current assignments, etc. So in this week, actually, this year's and previous year's assignment was fairly similar. So we do not have many new questions. We'll be discussing those questions by themselves. Maybe they are just framed a little different, maybe one or two questions, right? And the second part, what we'll do is like that unit one that you have gone through, the unit one courses that you have studied, right? We'll be going through those concepts, and there are places wherein some concepts have been touched upon and not really emphasized on, because that is probably not how the course is designed. But since we have extra time over here, we'll be touching upon those topics also. We'll be understanding them in a little more depth. Right, so that is what we'll be doing in these courses, right? So there are four. It is a 4 week lecture. We'll have these live sessions weekly for 4 weeks, right? As for me, I missed my introduction. I am Arveen. I am currently a PhD scholar at Department of Biological Sciences and Bioengineering, IIT Kanpur. So I am from the same institute that Professor Nitin is. And I have actually taken this course myself as a student, as a first year PhD scholar when I first joined the institute. And it was one of the courses that I really liked. I hope you will also enjoy this course, specifically if you are going to, be in this field of neurobiology. So that is what I'm doing my majors in right now. So it is a very, very relevant field to me, which is why I really appreciated having studied it in my first year. Right. If we are through with how these sessions are going to proceed, maybe we'll just begin with the session by itself, right? Okay, just a moment. So I'll keep admitting people if they are joining, or if you get disconnected also, maybe I can admit you because I'm getting that in a pop-up over here. So that shouldn't be a problem, I think, right? Yeah, I hope you can all see my slide, right? It is still on the title slide right now. Uh, and I'll be switching it now. So I hope that is working for everybody. How we'll be going through the discussion of assignment is I'll present you with one question. Now, the first few questions from this is the week zero assignment questions, which are generally, a little general knowledge or to test uh, just to test you or just to make you curious. Those questions, right? And they also have options over here, right? So first we'll read the question. I'll give you the options. You will have a moment to think about those options, which option you think will be the best fit for this. And then in the following slide, we'll discuss what the answer is. And if there is a logic behind that, we'll also discuss that logic as to why it is this particular option that is the answer. Right, so that is how this first part of the session will proceed. Yeah. So, which of the following structures is not present in the brain? Right. So, out of all the structures that are given, which one is not a part of the brain? Right. So, is it the cerebellum or the pineal gland or the hippocampus or the adrenal gland? Right. So, I'm sure even if neurobio is very, very new to you, and even if you have a basic understanding of biology from class 10, you will be able to get this question, right? Because this is talking about the major glands of the body, some of them, and you will recall from your class 10th, 11th biology where these are, right? So the answer over here is adrenal gland, right? So all these three, in fact, are a part of the brain. Right. So cerebellum is a part of the brain. It is just above the brain stem. Pineal gland is a part of the brain. It is near that region itself. Hippocampus is almost in the midst of the brain, right? It is controlling so many functions, be it the spatial location or the satellite cells are over here. It is also helpful in memory formation, right? All these things, these three are a part of the brain itself. The odd one out over here is adrenal gland, right? So, unlike all these other three, adrenal gland is actually not present in the brain. So, where is it present? So, it is actually present on the kidney, right? So, if you remember the structure of the kidney, there is this tiny gland on top of the kidney, and this is known as the adrenal gland, right? So, question number 2 was, which of the following statements is the most accurate, right? So, which of these statements do you think is accurate? Uh, Brain does not control the heart rate. Brain does not control the breathing rate. Brain does not control the knee-jerk reflex. Or brain does not control the vocal cords during speech. Which one do you think is the best fit answer? Right. Options are pretty simple, right? So that is why I'm not leaving much time. Yes, Jagrati, you may turn on your mic.
मैम एक्चुअली हमें आंसर टाइप करना है क्या?
Oh, right. So actually, this is a Teams meeting, right? And since you guys are externally joining this link, I don't think the chat option will be open to you, right? So it is not as freely functional as what is that other site that we have? Um, Okay, I'm forgetting the name. So Teams is not that friendly in terms of functioning unless and until I add your individual email IDs, you will not be able to actually answer the questions in the chat box. So it is, you can think about it. You can if you want, that is how we used to do it in the class. Actually, you are free to raise your hand and answer also. Otherwise, we'll just discuss this. There are about 10 to 14 questions, I think, this time. And then we'll be beginning with the lecture part of this series. So, just a moment. Mahavish, yes.
Yes, ma'am. The answer will be C. It does not control the nature because it is being reflex action then controlled by the spinal nerves.
Yes, yes. That is precisely the correct answer and the correct explanation for that answer also. So yeah, actually the pole function over here is not valid in Teams. Teams is the platform that our institute actually provides us. So this might be a better option for us to interact, right? You may raise your hand and you may answer along with the question, right? So yes, precisely what Mahavish said. It does not control the knee-jerk reflex, right? All other activities that are listed over here are in some way controlled by the brain, but the knee jerk reflex is not, right? It is one of the reflex actions, and reflex action is a quick action or a quick response. And for it to be that quick, it is actually controlled by the spinal cord. The motor neurons that are present in the spinal cord will actually control it, right? So that the signal is not transmitted all the way up to the brain, and then the signal is taken back from the brain to the part that is affected, right? That will just increase the time of this entire thing happening, right? So that is why the spinal cord is where the reflex action, such as the knee jerk reflex, takes place, right? So this is a typical diagram that you might recall from your previous, studies, if you had biology, of course, in your curriculum, right? So this is what the knee-jerk reflex looks like. The signal is actually taken across the spinal, across the from the muscle to the spinal cord, and in the dorsal root ganglia of the spinal cord is the cell body of the motor neurons, right? And these cell bodies will basically act upon, they will interpret the signal, they will tell what to respond in what is the response that is to be there for that signal. And then through different other efferent pathways, it will stimulate the muscles in your knee or in your thigh, basically to actually cause that jerk-like motion, right? Similar is the case for when you're touching a hot object, right? Your brain is not really thinking it. The signal is coming from your spinal cord. Yeah. So, which of the following scientists received a Nobel Prize for research related to the brain? Now, this is also something that we are eventually going to study during the course of this particular subject, that is neurobiology, right? You might not have come across it right now, but we are going to study the exact experiments also done by this Nobel Prize winning scientist very soon, right? So over here, the answer is actually Eric Kandel. Right, he is one of the persons who was working with the squid giant axons, right? And fabulous experiments he has done, which we will discuss as in when the course, gets into that phase. And he, along with Paul Greengard and Arvid Carlson, basically won the Nobel Prize in Medicine or Physiology in 2000, right? Yeah. So this is a very, very simple question. Does a mosquito have a brain? Is it true or false? Yes, Mahavish.
It's true. Mosquito has a brain with around 2000 nerves or 1200. I don't remember it.
Yeah. So mosquito, like other arthropods also and other insects also, does have a brain, right? So it is actually one of the study model organisms for olfaction based studies or neural network studies, which is something that Professor Nitin also does in his own lab, right? So mosquito is actually one of the organisms that is widely studied specifically for these two things. Also there is another arthropod that is studied, right? Another insect that is studied and is a very famous model organism, which is very close when we talk in terms of evolution as compared to humans, of course, I'm saying. It is closer to mosquito. Do you know what that other model organism is? Okay. So even if you don't recall it right now, you might have heard of it. It is the fruit fly or the Drosophila melanogaster, right? It is another very, very, vastly studied model
Uh,
organism, right?
is it Dorsophilia?
Yeah, it's Drosophila melanogaster.
Yeah, yeah.
Yeah. So it is a fruit fly actually, right? In layman terms, it is a fruit fly. So yes, that is another in specifically in genetic based studies and in the recent past, even it is extensively being studied for evolutionary, developmental, cancer, biology, etc. Right? So it is one of the model organisms, and that is the fruit fly. Another insect apart from mosquito. So they do have a brain, right? A lot of brain-based studies are done on these organisms. So this is just a representative image just to show you like the different areas responsible for different kinds of function over here. So what are the names of these areas? There is the mushroom body, there is the optic lobe, and other areas, right? And what are they doing? So these numbered areas are actually performing vision or smell related functions. Seven number this what is it? The SEV, it is performing functions related to taste, etc. Right? So these different areas are involved with these different kinds of functions. Yeah. So then the question comes, which of the following organisms has the largest number of neurons, right? So it has purely to do with the number of neurons over here, nothing else. Yes, Jagruti.
Actually, human.
No,
Ma'am,
that is actually,
elephant.
it is the elephant, right? So again, this raises that question that Sir has also asked and gone through in his particular course. That is the number of neurons what is determining how intelligent an organism is. So we did land. If you have gone through his lectures for week one, you would have understood that that is actually not the basis of like the basis of which organism is actually more intelligent, right? So number of neurons has nothing to do with that. It is the elephant over here that has almost 3 to 4 times more number of neurons than a human, right? Also because it is that much bigger than a human, right? So the connectivity that has to be built is across that bigger body instead of this compact body, right? So that is also why it is elephant over here. That is the answer, right? Uh, yeah. So again, it has more to do with the complexity with which these neurons form a network among themselves than the number of neurons by themselves, which determines how intelligent an organism is, right? Yeah. So another question is does fMRI have a better temporal resolution than EEG? Yes, Gaurav.
Ma'am, false.
Yeah, that is not correct. So what do you mean by temporal resolution? Can somebody tell me?
Ma'am, it is basically the We can say it is basically the frame rate, maybe how
Yeah.
accurately.
Yes. Not how accurate, that will
Hmm.
be incorrect. But the frame rate is actually a better, way to say this. So temporal resolution meaning how quickly it is happening, right? So
Hmm.
temporally meaning depending on time. So how quickly something is happening, it will have lesser temporal resolution. More temporal resolution if something is happening in the span of milliseconds, and it will have lesser temporal resolution if it is something that is happening in a span of maybe hours or weeks or days, right? So fMRI does not really have a better temporal resolution than EEG, right? So EEG is how is it happening? It is happening via a cap sort of a structure that is placed on top of your head, right? So the electrodes are basically on the surface of your head. fMRI, on the other hand, requires, and it, okay, the two techniques are a little different also. fMRI is basically capturing the resonance from within the brain, right? So, for now, we'll not get into, yeah, yes, Gaurav.
Ma'am, please can you explain what an electrode is?
Oh, what an electrode is. So, yeah, that is a good question. It is something that is basically going to transmit the brainwave pattern, which is going to be in the form of magnetism into an electrical signal, right? So I have prepared a presentation wherein I am going to emphasize on different techniques, right? So because you have been shown this particular, let me just come to this, yeah, this thing, right? So this map had been given to you, but I have prepared a presentation wherein I will be talking about what per se is lesions, what per se is PT, what is fMRI, what is the difference between maybe fMRI and a normal MRI, these kinds of things, right? Over there, we can emphasize on it more, but for a basic understanding or basic outline, what is an electrode? It is something that is going to carry an electrical signal by itself to a detector, or it is going to convert a magnetic signal into one of an electrical capacity, and it is going to show that to the detector, so that in the detector we get some values that tell us what is the brain status or what is the activity status of the brain, right? So it is generally the electrodes that are implanted inside your brain or inside your skull. They are like small, thin, very fine needles, right? They look like that. But essentially, they are like kind of wires that are going to transmit the signal across that particular thing, that is the electrode by itself, and to the detector, so that we can actually see what is happening, right? The signals that are placed on top of your, yes, Jagrati.
Ma'am, actually, sorry to disturb, but actually, I have another question. That course
Yes.
has started or not actually?
Yeah, so it has started. So, in the beginning, that is what I was saying. That you have like each week, I think there is an updation of 1 week's lecture, right? So it is a pre-recorded course actually that Professor Nitin has already recorded, and he is uploading those recordings, right? So that is where you have to do the learning bit. The assignment actually is where you score if you want to, get the certification and all. So the assignment part is where you have to look for the scoring thing. And like if you are enrolling for the examination, then based on what has been taught to you in those pre-recorded sessions, will you be asked questions in your examination, right? And this is a
Okay.
separate, like it is a live interaction. If in case you have any problems with those sessions, right? For instance, you are not understanding what you know what is that experiment that Candle had done, or you are not understanding like what is the contribution of Golgi versus Kahal, right? And like what exactly is the inference we get out of what Golgi said, and what is the inference that we get out of what Kahal said. Those are the kinds of things we can discuss over here. Yes, somebody raised their hand. Yes, Gaurav.
Ma'am, can we get the sample papers of the exam? Maybe sample paper or anything like that?
So I will ask that to your other TAs, that is Smith and Webby, I think. And you can also drop that in the Google groups, right? The discussion forum that we are on. You can drop that over there also, and I'll also ask them. But like traditionally, generally, we don't have sample papers.
Ma'am,
So, but
so
it is
yeah.
going to be similar to your assignments.
Okay. Ma'am, so does it will it have more mathematical or numerical part or just the
So that
theoretical
is
part?
just in the unit 2, there is some part that is a little mathematical, right? But in case of this, MCQ, even that is not going to be a major portion of your course. You have
Okay.
4 units. 3 out of them do not have, I think, any mathematical related, questions. Just unit 2 has some, and that also, if you understand the underlying concept for that, you'll be able to solve the problems. So over here, when we have subjective questions, actually, sir is not that because we have people from both biological and engineering background over here at in the campus taking sir's class. So he is very okay with you not knowing maths or them not knowing bio, right? So in that case, if your answer is reflecting that you do understand the concept, that is it for him. But in case of MCQs, however, that is not going to happen, right? But the MCQs will be simpler also. So you don't have to worry about that much.
Ma'am, so paper will be MCQ based, na, or some
Yeah,
theory?
yeah. It will
Okay.
be MCQ based. I don't think there will be subjective questions, but that again, once we'll discuss with your current TAs,
Okay,
other
ma'am.
than me.
Thank you. Thank you, ma'am.
Yeah. Yeah.
Ma'am, and is there in negative marking questions?
In your final assessment, there might
Yes,
be.
ma'am.
There might be. But again, so this is something that I don't think till now it would have been submitted, the exam that needs to be conducted. And I don't know how frequently it is changed, like the outline of that, how frequently it is changed. And you do have 2 new TAs, so we'll discuss it with them first. I don't want to say something that is not going to be true later.
Okay, ma'am.
Yeah. Yes. Yeah, as for this question, another very simple question, which of the following is not a part of a typical neuron? So is it the nucleus, axon terminal, microglia, or myelin sheath? Yes, Gaurav.
Ma'am, microglia?
Yes, microglia is not a part of the typical neuron, right? It is a different class of cell. It comes under the glial cells, which are they were thought of the supporting cells for the neurons, but now they do have some different functions of their own, right? Microglia particularly are the immune barriers in the brain, right? So they are the resident macrophages of the brain, and they have functions to do with the immunological aspect. Yeah. So you can see a typical structure of a neuron has the cell body, it has the cell membrane, the dendrites, axons. This portion where the axon is basically initiated, which is known as the axon hillock, oligodendrocytes, the nodes of Ranviers, the myelin sheaths, axon terminals, and the synaptic end bulbs. Right. So 3 of these are a part of the typical neuron structure, but microglia is not, right? Microglia is a different cell type altogether. So if a person suddenly comes across a lion in a forest, the autonomic nervous system that is likely to get activated is the sympathetic nervous system. Is this statement true or false?
It's true. The
Yeah,
statement is
True,
it is
ma'am.
true. Yeah. So, what is the difference between the sympathetic and the parasympathetic nervous system? Certain characteristics that differ, or certain
true.
responses of our body that differ? Okay, for instance, what happens to the heart rate?
Fight, it is in case of fight or flight situations. Sympathetic
Yeah,
nervous systems, and when we get into our relaxing normal state, it is parasympathetic nervous system. Like this is the
yes.
basic idea I have.
Yeah, yeah. So, like for instance, there is increased heart rate in case of our when we are scared, right? And when the sympathetic nervous system is active. But when the counter active system is going to get activated, which is the parasympathetic nervous system, its function is to basically relax us, right? So we will not like our heart rate will drop, right? There will be less palpitations into our brain, maybe. There will be more blood flow to our stomach so that digestion can be done. Because temporarily, that is not where the blood flow will be concentrated, because that is not what is going to be important, right? In case of a, fight or flight situation. Yeah. So, which of the following best describes the location of the primary visual sensory area in the brain? Right. So, again, this, if you have gone through the week one lectures and you have a fair idea of where everything is, you will probably get it. I will give you a hint for this one. So, is it the anterior dorsal or the posterior ventral or the ventral lateral or the dorsal medial? So somebody raised their hand. You are free to answer. I can't see the name over here. Um, the uh, kind of hint is yes, Gaurav, you can answer. Let me just give
Ma'am,
the hint, right?
yes, ma'am.
So the hint is that if you know where the occipital area of the brain is, right? You have studied the major areas of the brain. So this primary visual sensory area is in the occipital region of the brain, right? So where that is will be the correct answer. Yes, Gaurav.
Ma'am, and the visual cortex, maybe?
What?
Visual cortex.
Um, yeah, but where is the visual cortex also? And actually, the visual cortex is nearby the visual processing area, right? But yeah, it is in the same vicinity. But over here, that is not what is asked. The question is, the options are framed in a different way. So you need to know what is the dorsal part of your brain, what is the ventral part of your brain, what is the anterior, posterior of your brain, what is the central, medial, lateral, etc. Right? So this question is built to test that phase, right? So, for instance, just to revise, what is the dorsal part of your brain? It is this region, right? The area that is basically going to be on top, right? The ventral is the area just below it, right? So this area is going to be the ventral area, the one that is closer to my nose, right? Then the anterior will be in the front near the forehead. The posterior will be just opposite that at the back, right? Near the tip of the brain, kind of, or tip of the skull, right? Then the medial will be in the center. Like if this is the brain, then the middle portion is the medial one, and the lateral will be on the side near the ears, right? So, if you know where the occipital lobe lies, you will be able to answer this, is what I meant, right? So, yeah, let me just show it to you over here, right? So, this is the visual sensitive area. Which one? Okay, and it is sensory area. Sorry, I changed it in one slide. I did not change it in the other. It is a visual sensory area. You can also, so this is majorly the visual related area, right? For instance, Gaurav right now said the visual cortex, right? So it is in that vicinity only. That is also there in the occipital lobe itself, right? So we have studied these various lobes, the temporal lobe, the parietal lobe, etc. Right? So the occipital lobe is towards the end, right? And it is towards the lower part in that end, right? So this is anterior, that end is posterior, right? So it is in the posterior ventral end, right? It is not on the top of the posterior thing, it is towards the bottom of the posterior thing. So it is ventral. So posterior ventral over here is the correct answer. Right. So like if some other area has been asked, right, and you know for sure that that is in the temporal lobe only, right? So then where do you think or what do you think will be the response if suppose the area that was asked was residing in the temporal lobe? Then how will you say? In these terms, is it anterior, is it dorsal, is it posterior, ventral, lateral, medial? What is it? Something that is over here in this green area. Try, like, try to think like that.
Lateral will be there.
What?
Lateral. The position will be lateral.
Okay, so,
Ventral has
yeah, and
yeah, that's it.
yeah. So it is definitely ventral, right? We know the top of the head is the dorsal side and the bottom of the head is the ventral side, right? So it is definitely ventral. We know that, right? Temporal,
Lateral.
yeah. So that depends. We are not over right now. We are looking at from the side of the brain, so we can say that it is a lateral one. But the temporal region, a part of it could be towards the middle of the brain also, right? So, yeah, so that will vary, right? But definitely it will be towards the ventral region of the brain. Okay. So this and like what are the different kinds of brain sections that we have, the coronal, the horizontal, the sagittal, these things you can see once more, right? Those sections will actually also help you if you are ever in a place where you are sectioning yourself and you will know like why like in what kinds of studies I will take the coronal section, in what kinds of studies I will take the sagittal section, etc. Right? So I learned that during my PhD actually. Yeah. So, which of the following brain areas is most involved in keeping track of your location in the world, right? So, this again is something that has is a discovery that has recently also been awarded the Nobel Prize, not too long ago, not very recent, but not too long ago, right? Is it the amygdala or the hypothalamus or the medulla oblongata or the hippocampus? And I have actually already said it in this particular lecture also in the beginning. Okay, so I'll answer, right? So the correct answer for this one is hippocampus actually. Right, so hippocampus is the area that is, involved with the spatial, like it is keeping track of where you are spatially, right? And those new kinds of cells that were discovered within the hippocampus were the satellite cells for which the Nobel Prize was awarded a few years ago, right? So this is another very crucial area, right? And in a lot of brain diseases, specifically this, the dementia-related diseases, hippocampus is often one region that is very severely affected, right? Be it inflammation, be it dementia, it is very severely affected in most major, neuro degenerative disorders. Yeah. So in his famous experiments, Penfield measured the electric current generated in the brain when the patient moved specific muscles. I'm not sure whether this was a part of your week 1 lectures, but it was a part of the assignment last year. So that is why I've still kept this question. So is this statement true or false? I'm not sure if you guys have actually studied about Penfield as yet. So if not, I will just give you an overview of who is Penfield, what is the kind of studies he has done, right? So Penfield was a neurophysician, right? A neurosurgeon rather, right? And I think if I'm not wrong, he was a Canada-based neurosurgeon, and he used to treat epileptic patients, right? Now, what is epilepsy? Can somebody tell me? Does anybody know what is epilepsy?
हम
Like
मिर्गी का
and
दौरा
paralysis.
पड़ना.
Okay, 3 people responded. Now, one by one, you can respond. Let me just look at who you are. Uh, okay, Gaurav can go first. Yes.
मैम वो मिर्गी का दौरा पड़ना में भी इन हिंदी.
Yes. That is epilepsy only. And Ayush, yes.
Yeah, so it's basically like having seizures at like random time that can be triggered by any sort of stimuli.
Yes. So, do you know what is a seizure?
So seizure is basically like your whole body would seize, and there could be some movement of the body, and like the neurons basically they cannot transfer the data like properly through each other. So there is a
Okay.
disconnection.
Depression
Okay.
of oxygen
And
in the
who
brain.
else? Mavish, yes.
Oxygen circulation is also like depleted in that condition when the seizure attack takes place.
That is more like a stroke, right? It could be in case of a seizure as well, but oxygen depleted condition in case of brain, it is basically a stroke, right? Often the two accompany each other also. That is completely true. But if we are just talking about epilepsy, what it is? Okay. Does anybody else
Unconscious.
also want to add?
Paralysis.
Yeah, sorry, I did not get it.
Like and paralysis.
Oh, paralysis is actually very different. That is not exactly epilepsy. So, okay, I'll explain briefly. There are different kinds of epilepsy. The most common one that you might know of or might have come across, about sometime in your life or just heard about it sometime in your life will be the kind that I used to describe, right? That there is jerking movement and you are not able to control the movements of your body. There is something that is known as a seizure that is happening, etc. Right? So, and uncontrolled bodily movements are something that most people are aware of in case of epilepsy or in case of seizures, right? Um, Along with this, there are other kinds of seizures also. So this is what is known as the myoclonic or the tonic-clonic seizures, right? So there are different classes of seizures that are there, right? So myoclonic meaning muscle spasming that is not under your control. Tonic-clonic again, that there are other movements along with the muscle spasming that you are not able to control and that you are not aware of, right? And it is happening due to some thing that is going on in your brain. Now, there is another kind of seizure also that is there, which is the absent seizure, right? Now, what is happening in that? A person is just sitting, they might just be staring in blank space, and they might not be aware of it at all, or they will have no memory of it after maybe 10 minutes of the seizure also, right? So that kind of seizure is also there. It is not always the uncontrolled movement that is there. It is also like spacing out, right? Not like if I am spacing out, it will not mean that I'm having a seizure. Obviously it is linked to a particular kind of brain activity also, right? Now, what is this kind of brain activity? This is basically hyperactive, potentials that are going on, hyper firing of neurons in the brain will cause epilepsy, right? So that is also why it is very different from paralysis, right? Paralysis, the brain waves are slowing down a lot, right? It is going to a comatose-like stage, right? So that is towards the slower brain waves, the alpha beta brain waves, right? The slower brain, that is the slower one, right? I hope I'm not saying that incorrectly, the names of the waves, right? But it is going towards a slower brain waves. Whereas in case of seizures or in case of epilepsy, there is hyperactivity of these neurons, or misfiring of neurons is what it is called, right? So they are firing so much that no signal can be caught by the other neuron, right? Which is why there is uncontrolled movement that is happening, or there is uncontrolled things that are happening, right? Your mind is not aware of what is going on. It is just a misfiring kind of situation that is half-hazard kind of situation that is going on in the brain, right? So that is basically what is epilepsy. Now, why did I detour to that was that Penfield, yeah.
Can you please repeat the first type of epilepsy?
Oh, I said myoclon. I mentioned three kinds. There are actually more than that, but the three major ones, the two major ones that you will probably encounter is the myoclonic one, which has to do with the muscle tone. The tonic-clonic one, which again is very similar to the myoclonic one, and the third one that I mentioned is the absent seizures, right?
Thank you so
They
much,
are
ma'am.
also no problem. Yeah, in some cases, they are also stages of epilepsy, right? As it worsens, there different stages you can see through epilepsy, right? So, yeah, coming back to why we were discussing epilepsy. Now, Penfield was one of these neurosurgeons who worked with epileptic patients. Now, earlier, during the time that Penfield was actually working, and he was actually in medicine, even now, actually, a lot of it is still being followed. In case of epilepsy, it is not curable, right? It now, with some interventions, it can be managed, but it is still not curable, right? So it is that kind of a state. Like diabetes, for instance, is not curable. It can be managed to some extent, but it is something that is we don't have a cure for, right? Just a moment. Now, same kind of a thing is in case of the epilepsy also. So a very common solution that was given for epilepsy was lobotomy, right? Now, what is lobotomy? It is basically removal of the portion of the brain that is responsible for this misfiring neurons, right? Now, that will be different in different patients, right? If I have epilepsy, maybe I have it in the temporal lobe, right? If somebody else has epilepsy, they might have frontal lobe epilepsy, right? Some third person might have epilepsy due to some region in their amygdala, maybe, right? So the regions will differ, right? Okay, epilepsy itself is a very big subject, so I'll not get into details of that for now. So the solution for that, what the neurosurgeons used to do is they used to dissect or resect that area of the brain which was responsible for this epileptic episode, right? So that brain area needed to be removed, and it used to be removed. So it is like a cost to benefit, ratio. You will check that whether that brain area being there in the brain is more important, or whether the person not having seizures is more important, right? For instance, if that brain area is responsible for, say, if it is in the cerebellum and it is responsible for the heartbeat, or if it is responsible for the respiration rate, even if it is removed, the person cannot survive, right? So that is something that lobotomy cannot be done, right? But there could be some area where the person is suffering so much. So even if the person does not form long-term memories, it is okay. But we do not want the person to suffer anymore, right? So those kinds of things need to be weighed out, right? In case of lobotomy being done, and it should be very precise so that only that region is resected and no other region apart from that is resected, right? And the brain also has a little bit of plasticity so that even if a function is lost, sometimes it can happen that other regions of the brain can actually grow to learn how to perform that function, right? That is also always something that can happen. Brain is plastic, right? So plasticity is there in the brain also. So Penfield used to perform these lobotomies in his epileptic patients, right? So when he used to perform these lobotomies, what used to happen was first you have to search for the area, right? Now we have fMRI, now we have PET and all, right? So we can just through an MRI machine, we can just look at where the blood flow is there when a person is getting a seizure kind of episode, or just before the seizure, where is the blood actually focused, so that we can have an idea about the area where which is going to be the focal point of this epilepsy, where the epilepsy is originating, right? But earlier these kinds of sophisticated instruments were not there, right? So what these surgeons used to do was they used to have an open brain surgery wherein the person will be sedated, the skull will be opened, and inside the brain we do not have any pain receptors. I hope you know that, right? We do have it in the skull. Once the skull is open, inside the brain by itself, we do not have any receptors per se. Pain receptors, sorry, not we do have receptors. We do not have pain receptors, right? So over here, if my skull was open and somebody was poking in my brain, I will not feel any pain, right? So that is what used to happen, right? But so they needed to look, they used to insert electrode in that open brain, and they used to check where the firing is happening, where the misfiring is happening, where it is looking okay, where it is not looking okay, so that they can narrow down to the exact area that needs to be resected, right? So that is the kind of thing he used to do. And during this, he did not used to measure the electric current that was generated in the brain. Rather, what he used to do was he used to stimulate a brain region through a small electric current, right? So it is not like I am electrocuting somebody. It is not like that. It is like, for instance, to check your reflex, that hammer is, just below the patella region of your knee. It is just, bumped into it, right? It is kind of like that. So a little bit of electric current used to be, uh, there in that electrode, and it used to be stimulated. Different brain areas used to be stimulated to see, once this brain was opened, okay. Again, earlier there were not those many ethical committees that were there, right? For now, even if we have to work on mice, we need to get a lot of animal ethics approval, right? Human rights were not there that much, right? So once he was performing lobotomy on these patients, along with that, he was also a curious person, and thank God for him being curious at that time, because a lot of our understanding of which brain regions do what or are responsible for what has come from Penfield studies only. Okay, so what he used to do was that anyway, the brain is, this patient is on the table, and the patient, once the skull is open, is conscious. Okay, so the patient will be awake. In any brain surgery, actually, that is the case, right? The patient is always conscious after the this skull window has been made, right? So the patient used to be awake, and what Penfield used to do was, along with the looking for that area where which is to be resected, he also used to make notes, right? So once he's putting an electrode at one place, he used to see that what is the response the patient is getting, right? For instance, if he put it at area A in the brain, the patient would start laughing, right? Due to no apparent reason, right? But whenever he did that with any patient, right, if he used to put an electrode in area A, the patients always used to laugh. Means it has something to do with controlling laughter, right? If he used to, for instance, put it at some other area B in the brain, they used to, for instance, do something with their fingers, close their fist or something, right? So we know that that area is responsible for the motor functions of the finger, right? So those kinds of studies, he has made notes for those kinds of things, right? He has extensively studied it across his patients, and he has made notes that this brain area is responsible for this particular movement. This brain area is responsible for this kind of a response, right? So that is what Penfield did, right? So this particular, you will study it a little more in the course of this particular course, in this course itself, right? Apart from that, if you get a chance, you can actually also just watch or read papers or read articles rather. Papers are still difficult. Articles are even easier to read, right? So you can read those kinds of articles that are written on his experiments. They are very, very like, I think it will help develop curiosity, right? Sorry. Yeah. So as for this particular statement, it was false because he was not measuring the electric currents that was coming out of the brain of these patients. He was actually introducing or stimulating the brain regions with electricity of his own, right? Then bipolar neurons have two main dendrites and one axon. Now I have written it as axon. I want to change that right now only. Okay. I have given away the answer in between changing that. Anyway, so since I had already revealed the slide, it is actually false, right? So a bipolar neuron, as the name suggests, will have two major branches, right? Bipolar meaning two branches, right? So it doesn't have two dendrites. It has one branch that is a dendrite, one branch that is an axon, right? So unipolar is what? From the cell body, there will be one main branch that will, come out, and that will only have the dendrites also on one end, and it will have the axon also if it is required on the other end, right? The bipolar, as I said, will have two branches coming out from the main cell body. One branch will lead to the dendrites, other will lead to the axon, right? It will form the axon. A pseudo unipolar neuron is actually the one that I was mentioning earlier, that one branch will come out of it, and that branch can lead then into a dendrite branch and an axon branch, right? And multipolar neuron is the most common type that we have already always studied, right? It will have multiple dendrites that are going to come out and one single axon that is going to come out, right? Yeah. Okay. So another question. I think this part you have already studied. So, Golgi found that the brain is composed of different neurons. Is this statement true or false? So if you have gone through, I think, week one lectures, it is covered in that, as far as I remember.
True, ma'am.
So, Golgi, Cox staining, silver staining, that was not covered in week one lectures.
Ma'am, it is true. The answer is true.
Oh. No, actually, the answer is false. Okay. So, again, Golgi or Camillo Golgi, he along with Ramon Cajal also received a Nobel Prize for the discovery of these different neurons. Golgi basically what he did was Camillo Golgi, he developed a staining technique through which we could differentiate the neurons from the underlying tissue or the other things in the brain, right? So it was a silver nitrate staining, silver staining also we call it. One of the major salts that is used to stain it is silver nitrate, right? So these silver salts, when stain is made out of them and the brain sections that are taken are stained, uh, one can look at neurons, right? Now that is what was a contribution of Camillo Golgi, right? But when he did look at neurons, what did he say? What he said was something like this, like I don't know, freehand, will I be able to draw over here or not without a pen also? Yeah. So what he said was he found multiple, kind of structures like this and he drew them. At that time, we did not used to have those many photographs, but we did have microscopes, right? So what the scientists used to do was they used to draw, they used to look at the things in the microscope and draw it freehand, right? Uh, that is how we have wonderful, hand drawings of neurons by Ramon Cajal also, who has also used again the silver staining and looked at the neurons. So this kind of for instance, this is a region of the brain, say hippocampus. It is kind of like the C and V shape, hippocampus. So this is the C region of the hippocampus and he drew neurons like this, right? Camillo Golgi. And what he said was that once he looked in the microscope, he saw that there was a single network of neurons that was a single interconnected network of neurons that was going, right? So what he said was that all of them were the same neuron and they were interconnected, right? So that is why Golgi basically said that there was one interconnected network of neurons, right? He did not say that there were different kinds of neurons that were actually present, right? Now, who actually said that? Using Golgi's staining techniques, Ramon Cajal, who was another scientist, was he a doctor also? He was one of the scientists only, right? And I think a physician also. He also stained thin brain slices, and then he looked at them and he basically discovered those different kinds of neurons, that there are different neurons. There are pyramidal neurons and, uh, what are the different kinds of neurons? I am stumped right now. Uh, okay,
Motor
can't recall
sensory.
for the life of me. Yes, please help me. Yes.
Is it motor sensory in that way?
Sensory
These
neuron,
kinds.
motor
Not,
neuron.
not sensory and motor. That is a different kind of classification, like how they look. So in different regions of the brain, for instance, when we are saying, there are different regions of the brain, such as hippocampus and such as cerebellum, such as midbrain, such as cortex. So how are we classifying them as different regions? Just by their functioning? No. Because they also have some different kinds of neurons that are there, right? So, that is what I'm saying. For the life of me, I can't remember the other term that was there, right? Um, so there are different shapes of neurons, right? I can draw it even if I can't remember it right now. Yeah. So, for instance, some neurons might look like this. Okay, and this is where the axon is, and then the
Ma'am,
synapses and all. Yeah.
bipolar neuron, oligodendrocytes, and something like that.
Oligodendrocyte is not a neuron,
Ma'am,
actually. Bipolar is also different.
pyramidal cell or
Pyramidal
pyramidal
is
cell.
what I remembered. What is the other kind is what I'm not remembering. Right? It is based on how
Purkinje.
Purkinje. Yeah. For instance, Purkinje fiber. Right? So that is a kind of neuron. Right? So this is based on how this neuron is going to look, right? For instance, it was called a pyramidal neuron because the cell body was kind of triangular, right? In case of Purkinje, the cell body was more roundish, and the dendrites were like trees.
Yes, ma'am. Yes, ma'am. Tree. Yes, ma'am.
It looked kind of like a turnip head, right? So that is what Purkinje looked like. So based on how these neurons looked, uh, Ramon Cajal basically characterized them as different neurons, and they are indeed different neurons, and they have a little different characteristics also of their own, right? So there are different kinds of classifications of neurons also. In any case, there will be different kinds of classifications. Like you all mentioned that unipolar, bipolar, multipolar, that is a different kind of a classification. It is also for the neurons. For instance, I might say that if I want to classify colors, for instance, right, I might classify as brighter colors or dull colors, right? So I'll keep like this red, yellow, pink in brighter colors, and this black gray in white in the dull colors, right? Or I might also classify it as the primary colors and the secondary colors, right? So that the blue, red, then again yellow, this will go in the primary colors. This green will go in the secondary color, purple will go in the secondary color, pink will go in the secondary color, right? So there are different kinds of classifications. So don't get confused with that. Yeah. Okay, I'll just switch back to laser pointer. Okay, I have also taken over 1 hour in this part itself. So I don't know how much we'll be able to discuss regarding the other thing. Yeah. I think this is the final question. Which of the following techniques has the best spatial resolution? Is it the functional magnetic resonance imaging, or patch clamp electrophysiology, or electroencephalography, or positron electron emission tomography? Yes, Gaurav.
Ma'am, it is patch clamp,
Yeah,
I
it is
think.
patch. Yes, yes. It's correct. It is patch clamp. Yeah. So over here, we were earlier looking at temporal resolution, right? Temporal resolution, how did we define it? I told you that if it is in milliseconds, it is a better temporal resolution. If it is in days, it is a worse temporal resolution, right? So it has something to do with time, right? In case of spatial resolution, it has something to do with how close we can look at it, right? For instance, if I want to kind of how to explain. If I want to look at some part in on Mars, right? And I'm an astrophysicist, for example, right? So what I'll do is I might look through the telescope, right? That will have less spatial resolution because I'm sitting at my desk and I'm looking through the telescope, right? So that has lesser spatial resolution because it is very far away from the action spot where I want to look at the thing, right? If I ask somebody at the International Space Station to basically use their telescope over there to look at that particular spot which I want to focus on, right? That will be a better spatial resolution as compared to when I was on Earth and on in India and looking at Mars, right? And what if I launched a satellite and it went on Mars and it had a camera on it, such as a Mars rover, right? And that camera can transmit, signal to me, and it can show me exactly on that spot what it looks like. Right, it will be definitely much better than what I can see sitting over here, right? So that is the best spatial resolution. So it's likewise in the brain, right? You want to understand how the brain is functioning, how the neurons in the brain are functioning, etc., right? When you are looking at EEG, you are looking from the top of the brain, right? So it will not it will not have that good a spatial resolution because you will not probably be able to narrow down the exact area that you are looking at. Yeah, Gaurav. you are not unmuted if you want to say something.
Ma'am, does it also have a good temporal resolution, like both spatial and good temporal resolution?
It has a better temporal resolution also, but uh, it is also very invasive, right? So that is where it is lacking.
Okay.
That why will we do something like we might opt for fMRI, right? It has an okayish temporal and okayish spatial resolution, and it is almost not invasive at all, right? And this is very, very highly invasive. So, like, again, it is that cost to benefit ratio. What is it that you are studying? In case of research, obviously, we would want to look at single unit or patch clamp or light microscopy, right? But in case of diagnosis, for example, for a patient, we would always prefer maybe fMRI over PET, even though it might have a little less a spatial resolution than a single unit patch clamp also, but it is much better because over here there is nothing that is invasive. So based on these things, along with how invasive it is, we okay, I think there was something wrong. We basically look at which technique to perform. Yeah. So that is the end of the first part of the presentation, right? I'll switch to the next one. Uh, I don't remember how long it is. Let me see. Okay, so we'll try to cover most of it. Whatever is left, we'll pick up in the following lecture, the one that we have next week, and we'll discuss it again. We'll pick it up from where we leave today, right? Yeah. So this is more of a discussion thing. If we have been having a discussion anyway, right? It is to emphasize a quick recap of some of the concepts that you have studied and also to emphasize on some of the concepts that have been touched upon in your course, but have not been explained particularly because of the length of the course, right? Yeah. Okay. So when we talk about the human brain, right, some basic general knowledge about it. What are the basic, components of the brain? Something that we have studied from our school days, right, since middle school maybe. It is composed of the cerebrum, which is the major, bigger region of the brain covering the most area, the cerebellum, and the brainstem, right? And it roughly weighs about 1.5 kgs, right? Now, obviously, it is going to differ in everybody, but roughly, right, approximate average weight is about 1.5 kgs, right? Also, another interesting thing I want to tell you. So, cerebellum and cerebrum, you can see the difference in size over here, right? But cerebellum actually has almost half the number of neurons that are present in the entire brain. That much concentrated neurons it has over here, right? That is also something I found out much later. Yeah. So this is a quick timeline of what is happening when, right? So that again, like for instance, if we had done this earlier, you would have already known the answer to this Camillo Golgi question, or I don't think any other we have seen over here. So in around 500 BC, the Alcmaeon of Croton said that brain is basically the seat of mind, right? Then there was this brain mind hypothesis that kept going on. Then in 400 BC, Hippocrates, who is also known as the father of modern medicine, agreed that the brain was the seat of intelligence, right? So they had narrowed down uh, uh, to the brain as being the seat of intelligence and at how basically we are the creatures that are most intelligent in our environment, right? More than other organisms around us. In around 350 BC, Aristotle believed that the heart was the seat of intelligence and brain was a cooling mechanism, right? How the cars go, right? So there is an engine and there is a coolant, right? So Aristotle incorrectly actually believed that heart is what is making decisions and brain is just a cooling mechanism. It is providing that kind of a function, right? Like kidney is basically clearing blood for us. Similarly, brain is acting like a coolant is what he thought. But he was of course wrong. In around 200 BC, over here in India, Charak basically said, if brain is injured, many functions in body can be compromised. Right now, this is a natural observation, right? But at that time, we tend to miss a lot of natural observations also, right? That is why people who first discovered maybe the sun or discovered the moon, they also have a place in history, right? Because people tend to miss even the what we think is a common sense, right? But it might not have been apparent until it was told to us, right? Yeah. Then in 150 AD, Galen said that spinal muscles control specific muscles. Cerebellum controls muscles, cerebrum processes the senses, brain functions by movement of animal spirits through ventricles. So he got most of it correct, except for the part with the ventricles, right? Now, what is ventricles? I hope you all know this by now. It is the when you look at the sectioned brain, or when you look at um, this autopsied brain, the empty region in the brain is actually the ventricle, right? Now, because it was empty, he thought that that was where animal spirits were moving, right? But he was incorrect. Why is it empty? Because in the autopsy brain or in the sections, because that is actually the place where cerebrospinal fluid is flowing, right? Once the brain is taken out of its place, the cerebrospinal fluid is not retained, right? So, in the 16th century, Andreas Vesalius basically identified many structural features of the brain and concluded that brain functions through various nerves. Right now, he was also correct. In 16th century, Leonardo da Vinci made detailed sketches of the brain. Right. In 16th century again, René Descartes basically who was also he's a mathematician also. He was a proponent of dualism along with being a mathematician. He was a proponent of dualism of mind and body, and he said that these are two different entities and they interact at pineal gland. Right? Why did he say that they interact at pineal gland? Because in this autopsied brain that he saw that like whatever part of the brain is there in the left side, it is also exactly the same in the right side, right? So there are two of everything. But what he thought was that this nut-shaped gland that was there, which is almost in the middle over here, he thought that that was the only single unit. So that is what is controlling the entire thing, right? But in fact, that is also divided into an identical left and right half, right? That is also not true. Just because it was small and it was in the middle, he thought so. Then pineal gland controls movement of muscles through CSF in the ventricles is what he thought. And then in 1718, Luigi Galvani. Now this is another person that you will study about his experiments in detail also. He showed the role of electricity in nerve conduction and muscle activation, right? He was a person who basically looked at that electric stimuli of the frog, right? The frog was open, and even after it was dead, there was some electric. He could, make his hind limb jerk. This was the guy, Luigi Galvani, right? Then in 1850, Helmholtz, who basically said neural activity is electrical in nature and designed experiment to measure the velocity of signal propagated along the sciatic nerve of the frog, right? So he also carried forward this experiment and it was also true, right? But along with electrical, it is sometimes also chemical in nature, right? That is a disclaimer over here. Then in 1873, Camillo Golgi developed the silver salt-based staining technique for the neurons and claimed that the entire brain is a single large network, right? He did not distinguish between neurons. He said that the entire brain is a single large network of neurons, right? Interconnected neuron. In the 19th century, thereafter, Santiago Ramon y Cajal was able to visualize various different cells in the brain, which were different types of neurons, right? Some of which we discussed earlier. Then this neuron doctrine was given. Now, what is the neuron doctrine? It basically says that the nervous system is made up of discrete individual cells, which are the neurons, which are supported by astrocytes and by other glial cells. However, over the years, this neuron doctrine has changed, right? So over here, what is the center of everything? It is neuron, right? But over years, we are also now learning that glial cells are actually very, very relevant to the brain also, right? It is not just the neurons, but also the glial cells. If they were not there, neurons will not be able to function on its own, right? Yeah. So, these were two things that Ramoni Kahal eventually proposed, right? First, just by looking at those silver stained brain sections, he was saying these things, right? He has not performed any other experiment to prove this, but just based on those sections and looking at those sections in the microscope, he has made these observations. He has proposed these doctrines, right? First, he said the principle of connectional specificity. What does it say? It says that the nerve cells do not connect to each other randomly to make a network, but the connections are very specific, right? So it doesn't mean that just because something is lying in the vicinity of one neuron, it will automatically connect or form connections with that neuron, right? That is not how it happens. It forms a very, very intricate network, and there is some kind of, patterning that has gone to forming this network. Then there was principle of dynamic polarization. The electrical signals within a nerve cell flow only in one direction from receiving sites on the neuron, that is the dendrites as we know it now, to trigger region at the axon, which is a synaptic end pulse. Right? So just without performing any functional studies, just by observing, these slides, he was able to infer two things that are true till date, right? Both of these are true till date, right? There have been multiple validations that have been performed for these, and both of his observations were spot on, right? Inferences, observations, and which led to these inferences, right? Yeah. So then again, when we talk about the human brain, these are some facts, right? You may not remember them, not a problem at all. Just for the sake of discussion, we have around 86 billion neurons, right? And we have similar number, if not more, number of glia, right? So a number of glia might actually be more than these, right? These 86 billion neurons form around 10 raised to power 14 synapses. There are multiple neuronal cell types. The capacity of the brain of this Homo oh God, I am stumped today. I have gone back in time. I'm thinking about Homo erectus and Homo habilis. Homo sapiens. Yeah. That is around 1400 G, right? If you do remember this evolution, you will remember that the brain capacity kept increasing, right? As we transformed from our ancestors into the Homo sapiens that we are today, right? With Homo sapiens sapiens, actually, as evolutionists like to call it today, right? So more surface area was basically presented due to the presence of sulci and gyri. Even though earlier our ancestors had broader foreheads and a protruded skull and everything, but why do we still have more capacity than them? Because of the presence of sulci and gyri. Yes, Gaurav.
Ma'am, what is Salka and Gairai? What is this?
Oh, it is the folds that are present in the brain. So, like, just a moment, I'll just draw it. Like, for instance, you see, no, the brain, like, it looks like this.
The grooves.
Yeah,
The
the
But
those
grooves.
guide is saying that cerebrum has an like up and down structure.
Yeah,
Gyrus.
yeah. It is present throughout the brain, not just in the cerebrum only, but it is more pronounced over there. Yes. So this protruding part is the sulci, and sulcus. Singular is sulcus, and plural is sulci. And this, this part that is depressed over here is the gyri, right? So this is the sulci and gyri. I can, this one. So this protruding part is the sulcus, this depressed part is the gyrus, right? Yeah. Then comes phrenology, right? So it was a pseudoscience. It is not true, but it is something that people tried for some time, right? So it is kind of like from your facial features, I can judge what kind of a person you are. It is kind of that kind of a pseudoscience, right? So the study of conformation of skull as indicative of mental faculties and traits of character, especially according to hypothesis of Franz Joseph Gall, a German doctor, and such 19th century adherents as Johann Caspar Spurzheim and George Koch. Right, so they were the major proponents of this kind of pseudoscience that is known as phrenology. Now, what it majorly meant was that okay, this part of your skull is bulging, means you know you have this kind of a trait that you have aspirations, right, such as sublimity, right, or you are very very cautious, right, because this part has a dent, this part of your skull has a dent, means that you are not a very faithful person. These kinds of things is what this kind of pseudoscience actually propagated, which is not true, right? The surface of the brain cannot really account for what is inside, right?
Ma'am, this is not even a science.
Yeah, yeah. That is why it is known as a pseudoscience, because during the development of understanding as we have it today, it was a part, and a lot of people actually believed it at some point, right? So it it has been deemed as pseudoscience, not science, of course. Yeah. Then this methods in neuroscience, right? So overlay of this, we have already discussed it today, right? We'll just quickly go through it so that we can come on to discussion of the individual, kinds of methods by themselves also, right? So we have already discussed what is temporal resolution, right? So that is along the x-axis. What is spatial resolution, which is there along the y-axis. And the color coding that is given is that the colder colors are towards non-invasive or less invasive, and the warmer colors or hotter colors are towards invasive, right? So lesion, it is, if lesion is used for study of brain, it is invasive, right? Why? Because again, this is kind of like lobotomy, that lesion has to be removed so that the person will have some kind of a disability due to the removal of that particular region of the brain, and from that disability you will learn that okay, now this person can't form words, so means this area of the brain that has been removed was responsible for formation of words, right? So it is invasive, right? So these are the different kinds of techniques that are there, right? So we'll discuss them a little bit. So when we are talking about lesions and micro lesions, what does it mean? So over here, is an example of language mapping actually. So I will give you these slides also. You can go and you can look at these references, right? So I have always put a reference whenever I have put an image or anything over here. Or anyway, if you don't want to read a book chapter or a paper per se, just go and read, on the Wikipedia about it. Even that much knowledge about a new subject should be okay, right? And it will be in much simpler terms also. It must be written over there, right? So for instance, this kind of a technique was used for language mapping of the brain, right? Lesions and micro lesions. Micro lesions is just smaller lesions, right? So what does it mean was that people who had lesions in the brain, for instance, if you have a benign tumor, right, the tumor is removed, right, so that it doesn't spread, right? If you have kind of a lesion in the brain, it is a benign thing or it is a dead region of the brain, so that a small part of the brain is actually removed, right? Or even if it is not removed, it is anyway not functioning, right? You have a lesion over there. That is what it means, right? So in these through the study of multiple people who did have such kinds of brain lesions, the language mapping was done. What does this language mapping tell you? It basically says that if the lesion is there in this particular region that is basically marked by this arrow, right? If this is the area that the lesion is there in, it will basically lead the person who has this lesion to, have speech arrest during counting, right? So when the person is counting, they will be stumped. Like I was not able to look for that other kind of neuron at that time, right? It was just not coming to me. So worse than that, right? So they will just be stuck, right? Um, they'll not be able to count, right? So that area was actually, these areas are named after the scientists who have discovered them and discovered their purpose or functioning, right? For instance, another set of patients who had a lesion in this particular area over here, which is now known as the vernix area, if they had this kind of a lesion, then the literal paraphasic speech error during picture naming will happen, meaning again, it is a language based error, but over here they were only arrested when they were trying to speak out numbers, right? But over here, what will happen if they are looking at something, they will not be able to describe it. They might think correctly about it. They might think that they are saying it correctly, but they will not be able to say it out loud so that we can understand, right? And then this third kind of area, if the lesion is over here, right, then the person will have a difficulty in word finding during picture naming, right? So they might be able to say that, okay, this, like if I have a bottle in my hand, right? I will not be able to say that it is a bottle, right? I will not be able to find the right kind of word, right? So it is kind of like a library in our head also, right? Some places fetching the correct word, some places speaking the correct word, some places basically putting these words together, forming a sentence, some places actually responsible for the vocal cords for them to function, right? The correct kind of vibrations to happen so that the voice will come out. So these different areas have been mapped out by studies in people who had these kinds of brain lesions, right? And again, this is something we have known through fMRIs, right? After that, just to validate that whether this area itself is responsible for this kind of function, there have been intraoperative stimulations that have been done, similar to stimulations that I was telling you earlier that Penfield had done, right? So while operating, if somebody does stimulate that area, does it lead to speech arrest, or does it lead to a person having flawless speaking skills, right? So those kinds of validations can be done, and they have been done to basically tell us that, okay, this area is responsible for this thing, right? So brain is actually a very, very complex organ. We still don't know a lot of functions of the brain or a lot of regions, like which function is performed by which particular region. Maybe on a bigger, perspective, we can say, yeah, this thing is performed in the occipital lobe, this is in the temporal lobe. But there might be some other place outside of the temporal lobe doing the exact same function or performing a small bit that is responsible or contributing a very important part into that function that is already going on, right? So these kinds of things are already there. We don't know everything about the brain. That is why neuro is that much more fascinating, right? Yeah. So these are the examples of lesions, right? So if a person has a lesion at place X, for instance, what is going to be the response or what is going to be the pathological condition of that person, right? So that will help us in uh, basically looking at what is happening, right? So over here, this paper was titled A Human Memory Circuit Derived from Brain Lesions Causing Amnesia. Right, so these were different patients. These are brains of different patients that had amnesia. What is amnesia? It means basically that forgetfulness, or you don't have the memory of the past, or you can't form new memories. Right, different kinds of amnesias are there. Right, so from different brains had they did not have lesion at the same place due to which there was amnesia. They had these lesions at different places due to which different kinds of amnesias were also there, right? And from this we can say that, okay, from if there is a lesion over here, hypothetically, if this is the lesion, then it will not it will basically interrupt in formation of new memories for that person. That is the kind of amnesia the person has. If the lesion was in this place, it will make the person forget the basic life skills such as driving or cycling or something, right? If the lesion was in this place, for instance, it will, the person will not remember, the past, their past or whatever, right? So these kinds of studies have also been done through lesions and micro lesions. So it is a very gross invasive kind of thing, but if a patient already has a lesion, of course, then it can be studied to map out different regions of the brain and what these regions are responsible for doing, right? Yeah. Yes, correct.
Ma'am, is this dangerous than tumor or less dangerous?
Depending on, again, the region where the lesion is. What if the lesion is, at a place that is controlling your heartbeat? Obviously, it is more dangerous, right? If it is at probably a place that is responsible for fears, it just might make the person fearless. That is seriously not harming the person in that sense. So it is very relative in that sense.
Ma'am, but
Yeah.
what are the symptoms of it? Like, how do I know if I have it or not? How we can identify it? Like, I keep forgetting many
Yeah.
things.
No. So that is, of course, amnesia is not the same as forgetfulness, right? So I kept forgetting like so many things in today's class itself. That doesn't really mean that I have a lesion. It just means that I am not able to fetch the correct memory at the correct time, right? So again, diagnosis is tricky because we do not have the entire understanding of the brain, right? So that is why diagnostics is still building in this area, right? So how we can detect it is simply by CT scan. A lesion can come up in a CT scan also. A better, if you want to look at it in a better sense, you can look at an MRI. It will give a better, image of the lesion. So that is the basic diagnostic tool that is used. Um Through the behavior, I don't know if you have. I have had like elder people in my relatives or, in my friend circle, those families, wherein people have had amnesia. So it begins with forgetfulness, maybe, but you can tell that it is it is something pathological and something not that is not normal brain aging. You will be able to tell the difference because a person cannot forget how to brush, for instance. If a lesion is over there, if one day if you wake up and you say that I don't know how to drive, although you have drive for maybe 30 years of your life, right? That is something that is abnormal. So that is how it is generally caught, right? If it is at a place that is relatively harmless, it is relatively not affecting you that much, you will probably never diagnose it itself, right? There are some kinds of lesions that are just causing some kind of personality changes in people. If that personality change is so drastic, you might encounter it because you might go for a brain scan, right? But if it is not that drastic, somebody, you are just considering that somebody with time they have changed, they have just become bitter, right? And it is not affecting their personality so much, but it is causing a little bit of changes in them. You might not even catch it, right? Then we come to this other technique, which is a 2-deoxyglucose and the PET, right? So pretty similar they are. That is why I have kept them in the same bracket, right? So 2-deoxyglucose, obviously it is a variant of glucose, right? A variant that we can basically see through our diagnostic instruments, right? So it is developed by Louis Sokolov for laboratory animals initially, and it is an adaptation for PET scan that is done for humans and has provided direct demonstration of coupling between neuronal activity and glucose. Right. So what it means is, what is the base? Let's read this also, then I'll say. So 2-deoxyglucose technique is an experimental autoradiographic method that measures the regional brain glucose utilization. It can also be used to study neuronal activity and glucose coupling, right? So, okay, first off, what you need to know is that the only kind of, energy that our brain can utilize is through glucose. It cannot take up fats and convert it into glucose. It cannot take up, other energy sources and convert it. It needs glucose. Our brain needs glucose for functioning. That is why low glucose is harmful for us, right? So, this particular techniques, both these techniques, positron electron emission tomography and 2-deoxyglucose, both of them are based on the simple principle of glucose utilization by the brain. Right. So what it means is that if a brain area is more active, it will utilize more glucose. If a brain area is less active, it will utilize less glucose, right? And because we are introducing a kind of glucose that we can trace through a specialized kind of X-ray or MRI, we can actually see how much of it is being utilized in real time, right? So that is a outline of this kind of technique. So that is what happens in a PET scan, right? So, for instance, if a person is undergoing a PET scan, there is a normal machine which looks much like a CT scan or an MRI machine, right? There is a monitor, there is this rotating scanner that is there, and before that, this deoxyglucose is either injected or somehow introduced into their body, so that now the glucose that's going to go in the brain during the course of the scan, it will have this uh, kind of glucose that has this tracing ability that we can trace it, right? So this is what the outcome of a brain scan looks like, right? So the areas that are more active will show in hotter colors like orange, yellow, red, right? And the ones that are lesser active will show in cooler colors, green, blue, etc., right? So this is how deoxyglucose and PET scanning is done. So for instance, a person went in, and I think I might have kept another. This is a good example, right? So it is again from a paper, right? Over here you can see that there are two disease conditions that we have kept over here, the Alzheimer's disease dementia and frontal lobe dementia, which is also known as Pick's disease, right? And this is a normal brain activity, right? So age-matched control, we can say, right? So over here, you can see that there is consistent metabolic activity throughout the cerebral cortex, right? You can see so much of red, right? And it is there throughout the brain. So it will also depend on what you are doing, right? For instance, when you are listening to music, most of your brain is engaged. So glucose, you can look at, when you look at a PET scan, it will be throughout the brain almost, right? If you are, for instance, listening to something, only the auditory part will be engaged, right? So the place where the auditory cortex is there, only that place will light up, right? Most of the other part of the brain will not light up at all. So those kinds of studies have also been done using PET and 2-deoxyglucose to map that, okay, when we are for instance, blinking, which region of the brain is lighting up? When we are listening to something, which region is lighting up? When we are speaking, which region is lighting up? So that we can see that, okay, this region is responsible for this function, right? So now, with advanced techniques, this is also another thing that we can do. Over here, you can see that in case of both your pathophysiological condition, right? So all these people would have been in the same state. They might all, for instance, have been sitting and listening to music, and the brain scan will be done at the same state only, right? So in this case, you can see so much of brain activity. In this case, the brain activity is much reduced, and it is only there towards the front area, right? The back of the brain doesn't have that much. And this is frontal lobe dementia, which probably means that those neurons in the front area or the frontal lobe, they are already dead maybe, or dementiated over there, right? So the most of this activity is towards the back of the brain only, right? And it is much reduced as in both these cases as compared to the normal control, right? So this is a kind of diagnostic measures that we can take. And even in case of research, like 2-deoxyglucose, like it was said, it was basically for the animal studies, right? PET is mostly for the human studies or the diagnostic purposes, right? So these are the kinds of things it is used for. I hope it is clear. Yeah. So moving to fMRI, I think we'll just do fMRI and we'll end for today. Let me see what the other other upcoming slides are. There are a lot right now. So we'll pick up the others next meeting. fMRI we'll do for now, right? Yeah. One second. Yeah. So fMRI, what the F means over here is functional, right? MRI is normal magnetic resonance imaging, right? So functional magnetic resonance imaging is a kind of a MRI scan that shows which areas of our brain are most active, right? So again, the principle is very similar to PET. Now that you have understood that the region that is more active will consume more glucose. Similar to that, over here, what we are going to see is the region that is more active will consume more oxygen. Again, something that needs to be consumed, right, by all living tissues. So, tracking and comparing that activity to what you are doing, you were doing at that time, can help map your brain activity, right? Again, one of the things that I was mentioning earlier. It's most often used for planning surgery or similar procedures in the brain, right? So why? Because you need to assess that, okay, this part of the brain is where oxygen supply is very limited. So maybe this is the region where the problem is. So you can narrow down your search to that area. I can just create a small cranial window over here and just look in that area. I don't need to, remove my skull entirely and, go digging into my brain randomly, right? That is not what needs to be done now. A standard MRI scan uses an extremely powerful magnet, radio waves, and computer processing to generate highly detailed 3D pictures of inside of your body, right? An fMRI scan uses the same MRI machine, but is special because it tracks blood flow in different parts of your brain, right? So over here, blood flow is actually being tracked, right? So this is the difference between a structural and a functional MRI scan. So the machine that is being used is almost the same only, right? But over here we are just looking at the structure. Structure is definitely going to be exactly the same. But along with that, we are also looking at the blood flow to the brain, right? So it is a non-invasive technique for examining the brain activity also, right? While structural is just for examining the anatomy and pathology of the brain, right? For instance, we can see that, okay, lesion is there, right? Or, okay, this over here, ventricles look bigger, meaning dementia has begun. Over here, I can see some contrast. Maybe there is a clot over here, something like that only we can find out in structural MRI. Over here, we can actually see the flow of blood pattern. Yeah. So again, to map different areas of the brain also it is used. For instance, when you are speaking and fMRI is happening, these areas of the brain is where the blood flow is most, right? If I'm tapping my finger over there, right, like this, right? So this area of the brain is where the blood flow is the maximum. So this might be the one that is responsible for kind of, uh, motor functions for my fingers. Then listening. If I'm listening, these are the regions of the brain that are most active, right? Yeah. Okay. So we have three techniques that are coming up next. So this is where we are stopping for now, right? I just stop it. I stop sharing my screen. And this part we'll pick up in the next session now, right? As for today, till what we have studied right now in this lecture part, the second part, was there any problem with that? Like any doubt, anything, or anything you want me to add for the next session? That also I can do, right? I can prepare it beforehand. I can add it onto the slide so that it is easier to explain in that way. Okay. So if not, we'll end the meeting for now, right? I'll just stop the recording. I'll end the meeting. Thank you so much for attending.
Thank you so much,
We
ma'am. Thank you,
most
ma'am.
welcome. Most welcome.